Hey guys, today's lesson is about gene mutations. We've actually talked about gene mutations before when we were talking about chromosomal mutations like non-disjunction, but today we're going to get more in depth now that we actually know about nitrogenous bases and codons and that kind of stuff. So for today's lesson on gene mutations, we're going to go over the difference between somatic and germline mutations. We're also going to briefly compare and contrast chromosomal and gene mutations just so we don't get them confused. Then we're going to look at the different types of point mutation and frameshift mutation. We're going to talk about the types of mutations that can be both gene and chromosomal. And then we'll look at different types of mutations based on their results and the things that cause mutations. So first of all, just a reminder, somatic refers to your normal body cells that are diploid. Germline will refer to any of your germ cells, so egg and sperm, the ones that are haploid. So mutations can happen in any cell in your body. They can happen in the somatic cells, in which case they will only affect the person who actually has the mutation. Or germline mutations can also occur, but they are the ones that can be inherited by offspring. So just a reminder, chromosomal mutations happen when the number of chromosomes is abnormal because of things like non-disjunction during either anaphase 1 or anaphase 2. You can also have chromosomal mutations that happen when a big chunk of a chromosome is missing or damaged. And then gene mutations are the kinds that happen when within your DNA, the sequence of nitrogenous bases gets changed. So for example, instead of like ATA, you might have like AGA or something like that. So yeah, that is obviously going to change the codon when you change the nitrogenous base, which means that it has the potential to change the amino acid that's being coded for. So you could have a point mutation, which is also called a substitution, which is when you have just a single nitrogenous base pair substituted out. And that can result in either a silent mutation, a missense mutation, or a nonsense mutation. So a silent mutation is when you change the nitrogenous base. So obviously you have a different codon, but that codon, the new codon and the original codon both code for the same amino acid, which comes down to like the redundancy of the genome. The fact that there's a bunch of different codons that code for the same amino acid. So silent mutations will have no effect whatsoever because the protein's not going to change because the amino acid didn't change, even though the codon changed. Then you have a missense mutation. This is when the codon is changed and that new codon codes for a new amino acid. So instead of having like a methionine, you might have like a proline or a tryptophan. You're still coding for an amino acid, but it's a different amino acid, which means you will have a slightly different protein. So for example, sickle cell anemia is an example of a missense mutation. That's when people still have functioning red blood cells, but their red blood cells are a different shape than normal and they don't function as well. They get kind of clotty and they don't hold oxygen as well. And then a nonsense mutation is when the new codon is a stop codon. So you go from having a codon that codes for an amino acid and then the mutated codon codes for stop instead. Because of that, you end up not even making the entire polypeptide. It's not just that you have one new amino acid, it's that you're like missing a bunch of amino acids in your protein because you stopped making the protein too early. That is often lethal to the cell because it just can't function with that much of the protein missing. So those are the three types of point mutation, where you just sub out one nucleotide for another. And then you can also have a frame shift mutation, which is going to shift all of the nitrogenous bases from the point of the mutation onwards. There's two types of frame shift mutations, either a deletion or an insertion. With a deletion, at least one nucleotide has been removed from the DNA. So all of the nucleotides from that point onwards in the DNA shift forwards one spot, which means that all of the codons from that point of mutation onward are all wrong. So these are usually much more damaging than point mutations, because instead of changing one codon, you're going to change a bunch of codons. So for example, cystic fibrosis, which is a deadly respiratory disorder, is caused by a deletion frame shift mutation. And then insertion is when you have an extra nucleotide added to the DNA. Once again, all of the codons will shift backwards one this time. But again, from that point of mutation onwards, all of the codons are wrong. So your protein is being made very differently from how it should be with frame shift mutations. Huntington's disease is an example of an insertion frame shift mutation. Again, it's a deadly disease. Most frame shift mutations, again, are much more lethal than most point mutations, because instead of just changing one amino acid, it's changing a bunch of amino acids. You can also have translocation and inversion mutations. These are both examples of mutations that can affect both genes and chromosomes. Essentially, if it's a large chunk of the DNA that's moved to the wrong spot or inverted, flipped upside down, then it's chromosomal. If it's a smaller section that happens to, then it's a gene mutation. But either way, with a translocation, you have a part of the genome moving to the wrong spot. With an inversion, you have part of the genome flipping upside down. These types of mutations are more commonly chromosomal as opposed to gene. So if you're not told which one it is, assume it's chromosomal. So again, translocation happens usually during prophase 1 of meiosis, when the non-homologous chromosomes are crossing over. Again, if a big chunk of the crossing over goes wrong, that something ends up in the wrong spot, it's a chromosomal mutation. If a smaller chunk gets moved to the wrong spot, it is gene. Translocation can also happen during DNA replication, but that's much less common. And then inversion, again, it depends on the codon whether or not as a gene mutation an inversion would have any effect. As this example here describes, if a codon is palindromic, so it can be read forwards and backwards the same way, it doesn't matter if you flip it backwards. It'll code for the same thing. So these are just some examples of what mutations could look like. So here I've swapped out a U for a C. In this case here, I've swapped out a U for an A. Both of those are types of point mutations, because I'm just swapping out one nucleotide or one nitrogenous base for another. With this example here, again, that's another point mutation. Actually, no, it's not. This is an insertion, so a frame shift. So I've added in an extra C here. So all of the codons afterwards have got shifted down. With this one here, I have removed a letter. I've removed this U. So that's also a frame shift mutation. That's a deletion. And then in the last one, I've done an inversion. I've taken that entire gene and inverted it. So I would, if I were you, just practice going through these. Yeah, and right now these are mRNA sequences. So you can practice using your like mRNA codon chart to see what the amino acids would be in these cases. And especially for the A and B, the 2-point mutations, figure out if they would be a silent mutation, a missense mutation, or a nonsense mutation. So what you're going to be checking for in that case is to see if UAU and UAC code for the same thing or code for different amino acids, or if UAC or UAA are stop codons. Okay. As far as causes of mutations go, mutations can happen either spontaneously or can be induced. Spontaneous mutations are ones that take place naturally. They're caused by mistakes during DNA replication or mistakes during cell division. So that would be like, you know, the stuff that like DNA polymerase 3 and DNA polymerase 1 are supposed to just like catch during their proofreading, but don't always, or non-disjunction, that kind of stuff would all be spontaneous. And spontaneous mutations happen all the time. And then induced mutations are mutations that are caused by a mutagen outside of the cell damaging the DNA. These are a couple different types of mutagens. So physical mutagens will actually physically damage the DNA from a distance. So that's things like radiation, whether that be X-rays or gamma rays or UV radiation. All of those can pass through the skin, go into your cells, into the nucleus, and actually like damage the DNA and break the DNA. Then there's chemical mutagens, where that's where molecules actually go through your cell membrane into the nucleus and chemically react with the DNA, which causes it to change. So that includes things like cigarette smoke, gasoline fumes, defoliants, like things that take the leaves off trees and stuff like that. So that's like Agent Orange, for an example, which was a defoliant used in the Vietnam War to essentially kill the trees so that the Viet Cong couldn't hide in the jungles. Anyway, it's a horribly damaging mutagen. If you want to be horrified, Google it. And then teratogens, we've talked about before. Those are mutagens that can cross the placenta and actually damage the developing embryo. And then carcinogens are a specific type of mutagen that can also be either physical or chemical, but that specifically cause cancer by mutating genes that regulate cell division, like tumor suppressor genes and oncogenes. Okay, so that is it for today's lesson, a nice and easy one, but let me know if you have any questions.